looking at a story over the course of the weekend about a children's hospital right here in Dubai. It's the Jalila Children's Hospital, which has sequenced the genetic material of some of its young patients to help diagnose their illnesses and accelerate their treatment plans. The work at the hospital is thought to be the first use of rapid whole genome sequencing in the Middle East. That's according to officials. So scientists say they hope to use this technology more widely and to build up a database that could really help improve diagnoses for people, specifically from the Arab world and other regions. Because, of course, these things, when it comes to genetics are related to regions and ethnicities as well, and they're traditionally underrepresented in genetic studies. So this was all reported in the Journal of Genome Medicine, and the work involved sequencing the genome, or all of this genetic material, of 5 newborns and infants at their 200-bed facility. So to discuss this in a little bit more detail, we're joined now by Alan Taylor, who is genetic counselor at Al Jalila Children's Hospital. Good morning, Alan.
Good morning, Sonal. Thank you very much for having me.
Thank you for taking the time to join us. And I guess first, tell us a little bit about the science that goes behind genome sequencing. Let's keep it top level, of course, for our audience.
Yes.
But just give us a bit of an understanding.
For sure. I think as a genetic counselor, our job is to break everything down. So I'll give you, like you're saying, an overview of what genetics are. So when we start talking about what the testing is and the benefits of it, hopefully the listeners will have a better understanding of the benefit of this type of technology. So to give you a 101 on genetics, again, I'll keep it top level. The analogy I like to compare to our DNA is to a book. That if we think of what DNA is, I'm sure everyone's heard the term gene before, we can also think of a gene as a recipe. And if we think of all of our DNA in a body as a book, then this would be all the recipes of a human within a recipe book. So what the test is doing is that in the English alphabet, there's 26 letters. In the alphabet of DNA, there's 4 letters. So when we do this analysis, we're looking at all of these letters. So to give you a concept, there's about 2 billion genetic letters. So what we're doing in this is one aspect is looking for a spelling check. The same way if you type something in Microsoft Word, it's going to do a spell check and identify if a letter's out of place. So that's one aspect of this technology. And the other benefit of this technology is that, again, with the book analogy, a spell check wouldn't notice if a page was missing or if the chapters got rearranged in the book. So this technology, this whole genome sequencing that you mentioned, can both look at the spell check and looking for the chapters or pages mixed up in one assay. So that's really important because all the other technology that we have will either look at the spelling change or look at how the pages are missing or how the chapters in the book are muddled up. But this test can combine the two into one combined test.
I've never heard something like this explained so clearly. So thank you for that, because it is. That metaphor really does help explain what we can expect from it. But once we have this information, once we have this kind of book and this data, how does it actually help provide diagnoses when it comes to issues that people might be having where they're a bit difficult perhaps to diagnose?
So again, I have to thank you. You kind of introduced it very well, just the importance of understanding what normal genetic variation looks like. That when I'll speak a little bit in a couple of minutes about trying to understand the Arab population versus other populations, about why we want to do this research. So what we're doing, like I was saying, the spell check, is that first of all, we have to understand what normal genetic variation looks like. And this is why the whole world, we all look different, is because of small changes in our genes. What this test is doing is looking for a change in a gene that stops it working the way it should, resulting in genetic condition. And why this is important? Because if we have a diagnosis, then hopefully there is a treatment for it, and we can start that treatment very soon. And in a neonatal situation for many metabolic disorders, the sooner we have a diagnosis, the long-term implications are huge, are massive. So that aspect, having a rapid diagnosis in a neonatal ICU situation is very, very important. And also in situations where even if we may not change treatment, we stop unnecessary diagnostic work for the patient. We know what to expect for the future. And in a way can give parents some sense of closure that they know what the situation is and it can start making plans for how to help the patient in moving forward.
Can you give me an example of what this might look like in a patient case? So let's say you have a newborn baby, for example, who might be presenting with certain symptoms or issues.
Mm-hmm.
If you were doing a diagnosis, I suppose, in a more traditional method versus using this genetic method, what would the difference look like in terms of how this would be handled and how quickly we might get some answers? Because you mentioned there that it would do away potentially with the need for extensive testing.
So just focusing on rapid whole genome in particular is that it's an accurate diagnosis. Can't speak to a lot of the other testing because I'm not a doctor, I'm not a medical doctor, so I wouldn't be able to say specifically how the prognosis would go. There would be a lot of biochemical tests that would be done because what we're looking for focusing on one particular diagnosis of a metabolic disorder, the way present with seizures, some other complex situations where the child's not breathing and requires intubation. But there would be biochemical markers that like I was saying, the chemistry of the body is not looking right, that we're seeing an elevated protein or biochemical, that will give us an indication as a diagnosis, which typically that would be enough to make a clinical diagnosis with some other findings, but you would eventually want to do genetic testing to confirm this diagnosis and help the family maybe make plans for the future. Whereas with this, that we have the accurate diagnosis from a very early stage.
And so far, in terms of the research that you've done, you've looked at the genetic material of 5 newborns and infants. I mean, what kind of sample size do we eventually need to get to? What's the next step for this kind of research so that, as we've discussed, there is perhaps a bit of a dearth of information specifically on this region when it comes to genetic studies compared to some other places in the world? What can we do to bolster that and make sure that we do have enough information here within the region for the nationalities that are based here?
So that's a great question. There's actually 2 parts to that question. The first one is how much do we want to expand this project, having rapid whole genome, that we are planning on. We did 5 as part of an initial study, and we did have exciting results. We're able to prove that in an ICU setting, we're able to find the diagnosis rapidly. And in the paper that we published, it was an average of 37 hours from when we got the sample to when we got the results. Now, this is huge if you've never understand genetic testing, that whole genome sequencing, if it's not rapid and does standard, will take months, 2 to 3 months before we have a result. So having it within 3 days is a massive turnaround time. So with the first 5 that we did, we had a 60% diagnostic yield, which just demonstrate how important it is. And we found 3 different types of test diagnosis, which were very important to demonstrate. One was metabolic, one which was the sequence variant. 2 of the patients had that spelling mistake. And the third one was, like I was saying, the jumble up of the papers within a book. Now, we will like to expand it in the future, and we're hoping to expand that into many more patients, maybe up to 50. And at that point, after demonstrating the importance and utility of it, and then from maybe moving forward from there, rather just on a research basis, actually implementing this in a clinical basis for every patient moving forward. But again, the question always is about resources and the cost. But if we demonstrate the importance of it, then there's always a hope that we'll have funding for this, and insurance companies will understand the benefit of it as well, because they're always going to look at the cost benefit. So if we can prove that, then moving forward, we can do this on a clinical basis. Now, the second part of your question was, how can we understand populations that are underserved, like the Arab population? So the first thing, this will be a separate project, and I believe MBRU will be launching a project for Emirati genomes over the coming year. And the benefit of that is, like I was saying, before we can understand what genetic diseases look like, we have to understand what the healthy population looks like. And the only way to do that is to sequence the healthy population. And what we need initially is about 1,000. If you start off with 1,000, then you've got enough data to try and start extrapolating to what normal genetic variation is versus what disease variation is. So some of the big projects that happen are in America. So they've obviously very well studied the white population. But if we look at global controlled population databases, and just a controlled population is a healthy population, the Arab population is very underserved, and it is something that needs to be addressed moving forward so we can help people with Arabic backgrounds to help identify a diagnosis rather than doing testing and having no benefit from it, where we have this genetic change and we don't know whether it's a normal change that we've never seen before or it is actually a disease-causing change.
And it sounds like you're doing a lot to help address some of that challenge in terms of this underserved population. So thank you so much, Alan, for joining us today to discuss this.
Thank you very much.